Hello everyone. Welcome to the live sessions, first live sessions for the course Experimental Biotechnology. And if you have any questions, you can post your question so that we will be having a fruitful discussions. And since the session is very limited, only for 1 hour, so I think you all should take this as an opportunity to ask your queries or questions or anything which you could not be able to understand, and I'll be happy to answer. So good evening, everybody. So you can ask the questions. Now, Bhavneet is asking, sir, can you explain the electrophoretic mobility? Um, I think electrophoretic mobility is a very big topic. I think better we should discuss what you could not be able to understand. Okay. So you just write down what you could not be able to understand, Bhavneet, and we'll be happy to discuss that. Uh, uh, then she's saying that we have to detail instructions on how to handle the gel electrophoresis in lab. Um, okay. Dear sir, which one will act as a plunger in stacking gel of SDS page, glycine or tris SCL? Okay. So, uh, Hemant Nayak, it's a electrophoretic pressure what is going to be work as a plunger, right? So you have electrophoretic current which are coming from the backside, either it will be by tris act, and then you have a blockage with the help of the glycine, right? So because of that, because you already have electromotive force from the backside, and since but molecules will not cross the glycine because glycine is slow running, and that's why to be very specific, the plunger in this case is the ions what are moving on the backside. Um, then Hemant is also asking, you said that the CI molecule act as chloride molecules act as a plunger in week 1, lecture 1 video, but as glycine moves slower in stacking gel, will it won't act as a plunger? No. Plunger is something which actually pushes the content, right? So glycine does not pushes the content; it only stops the content, right? So that they should not run away. And so that's why I said, the ions are going to be the plunger. Sir, can you explain if I want to write a term paper, what procedural steps should I follow? Bhavneet, I could not follow your question. I think if better you explain me what kind of term paper you are talking about. Then Hemant is asking, SDS page protein moves through the pores of page gel. In agarose, how will DNA and protein pass through? In agarose also, the DNA pass through to the pores, but the pore sizes are pretty big compared to the SDS page. Uh, how will the DNA pass through? Okay, they pass through because they are, their size is smaller than those pores. So that's why they pass through from those pores, but still they are actually experiencing the friction. Uh, Sir, could you please explain the below question from assignment number 1? Okay. Wait. Okay. So let me explain you that. Okay. So if you want to design a solution for checking the stability of a drug in stomach-like conditions, the pH range you will maintain the in solution is so you're going to maintain the acidic solution because the stomach pH is acidic. So you are going to maintain a very low pH. Exactly don't know the option. So it could be somewhere around 2 or 1, whatever is been given. Uh, Bhavneet is asking about review paper. So, are you planning to say, are you trying to say that I should suggest you a topic, or you are asking me how to write a review article or something like that? Um, See, I really not sure what you really want to discuss actually. Are you planning, are you want to discuss, Pavneet, how to write the review article, or what are the components we should have when you want to write a review paper? So Darpana is asking how we can actually be able to detect the particular protein. So you cannot be able to detect the particular protein simply by the SDS page. You might have to, until unless it is actually overexpressing very well, until unless it is overexpressing and you know the molecular weight of that particular protein, it is not possible to detect that particular protein. The only other tool is that you can actually use the antibodies which are specific to that particular protein, and then you can use the Western blotting to detect that particular protein. Now Bhavneet is asking which matrix would be used as a stationary phase. So matrix remains the same, whether it is the cation exchange column or the anion exchange column. Mostly we use the Dextran matrix or Cepadex matrix, and I think she is also asked they want to discuss what are the different components are there into the review articles. Um, so the components, what we can do is we have to first have to have a abstract, right? So let me, I think we can discuss that since I now understood, right? Uh, so this is nothing to do with the course what we are discussing, but just to explain you, right? If you want to write a review article, for example, right? Let me give you an example so that you will about understand, right? Suppose I want to write a review article on coronavirus and its impact on society. Okay. Now, if I if I have to do this, okay, what I'll do is first I'm going to. So this is the title. Okay. So this is the title. What we are going to have, right? You can change the title and make it more catchy also. And then first thing is you are going to write the abstract. Initially, when you want to write the abstract, it could be a crude abstract, which can be refined later on depending on the type of literature what you are going to cover in that particular review article. But so if you want to write an abstract, you should like write a 200 words abstract, where you can just write a crude things that what are the things you are going to cover, or what your things you know about this particular aspect, right? And then you are going to talk about the introduction. So introduction, we can actually have introduction about the coronavirus. Then the we can have the disease biology. So when you talk about the coronavirus, you can talk about the different types of proteins and the code proteins and all that, right? So you can discuss all that, because that you will use later on for, saying that there is a detection technique and all that. And then you talk about the disease biology, then you talk about the pathology, something like that, right? And then ultimately you are going to say about the impact, right? So when you are going to talk about the impact, you are going to talk about the economical impact. You're going to talk about the psychological issues, right? Psychological issues, psychological impact. Then you're going to talk about the health, right? and and then you also going to talk about some positive stuff, right? For example, that how it has changed the infrastructure of the country and all other kind of things. So how it has changed the health infrastructures and all that. Now, once you have written the introduction, then you're going to take the topics, right? You're going to first write the brief introduction, and then you are going to take the topic which you are going to discuss in your review article. So I will start again the same way, right? I'll start with the coronavirus. But now what I'm going to do is I'm going to write in detail about this. I am going to write in detail about how the people have discovered the coronavirus and blah blah blah, like that. And then I'm going to end up with, what are different proteins, what are detection technology, and so on. And then I will elaborate all these topics in much in detail. I can add some figures, I can add some tables, uh, like that. And that's how, after that, you are going to have the discussion, right? So you can just discuss all these aspects, and later on, you're going to have the conclusions and future direction, something like that. Okay. So that is just idea to give you how to write the review articles. Okay, if you once you start writing, actually you will be going to be better. Okay. Now Hemant is asking about, can we conduct the SDS page in the horizontal directions? So my answer is no, because the one of the issue is with the uh, SDS page is that it should be discontinuous, right? It should be discontinuous, so that there should be, why it should be discontinuous, right? Remember that you should have, this is your SDS page, right? Where you have a stacking gel, right? And then you have a resolving gel, right? So, and these two gels, so there's a lower tank, right? And then there is a upper tank, right? And these two tanks are not connected, because the gel is connecting to them, and that's how the electrons or the ions are coming from this side, and then they are falling into this one, right? So and so this is a positive side, and this is the negative side, right? So and this is discontinuous system. This is discontinuous because the electromotive force what is required to run the protein requires to be very high, because the protein are very require a very high electromotive force, and that high force is only being generated when the system is discontinuous, so that the ions actually moves through the gel, and in that process only they actually take up the protein along with that. If you do the horizontal, right, suppose you design this like in a horizontal way, right? uh, even like, for example, there's no need to have a stacking gel, right? But even then, right? So if you do it in a horizontal direction, first of all, that discontinuous thing is going to be go, right? Because you are going to have the buffer on top of this, right? So the 90% of your current, or it will go on top of this gel. It will not go through the gel actually. And because of that, the proteins are going to migrate very less. Why you require a high electromotive force? Because the pore size is very small actually in the case of SDS page compared to the agros. Agros, the pore size is bigger actually. So even if the only 10% or 20% current goes through the gel, it will still be enough to generate a required electromotive force to to run the DNA. Is it clear, Hemant? Okay, now Darpana is asking, when we analyze proteins or DNA through electrophoresis, can we again obtain them for future use? Okay. Uh, when we analyze protein or DNA through electrophoresis, can we again obtain them for future use? Yeah, definitely. Why not? If you are running the proteins, or if you are running the DNA, right? You can very easily be able to collect both of them, right? If you are interested, you can do what you can do for the DNA anyway. It is easy, right? You can do the electroelution, right? You can do the elution, right? You can do the elution from the agarose block and you can collect, right? That is anyway. I think I don't know if we have discussed already, but I think that is what we are going to discuss, or we might be. So that elution you can do, right? If you got the DNA in agarose, you can do the elution and you can collect the DNA. For the protein, you can do the electroelutions, right? You can do the electroelution, and I think that we are going to discuss when we are going to discuss about the antibody production. So that time we are going to discuss about the electroelutions, right? What you are going to do is you have the protein in the block, right? That block you can just tie up into dialysis bag, right? And put this dialysis bag into the electrophoresis chamber with the buffer, right? So what will happen is the protein is going to run, and then since the block is going to be over, then it eventually going to fall into the this bag, and that's how you can be able to recover this protein, or even the all the protein also you can do that. Uh, then Pushpaja is asking, sir, can you explain once acrylamide polymerizations? Acrylamide polymerizations, what aspect you want to discuss? Just write me down. We will discuss that. Okay. Can you explain why the protein oligoesters cannot be determined using the gradient urea page? But urea page instead is used only for folding stages. Okay. 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 Good. So I think urea page. Urea page is, I think we last time also when I was discussing, I think some student was asking in the discussion forum also. So what we are doing, right, in a urea page, what you are doing is you are actually incubating the protein into a different concentration of urea, and then you are also using the different concentration of urea you're putting into the wells also. So you can imagine that I have like the 8 urea concentrations, right? And for every strip, right, this is the different urea concentrations, right? This is the different urea concentrations. So this whole strip is actually of that particular. So this is the no urea, this is the 1 molar urea, this is the 2 molar urea, this is 3 molar urea, this is 4 molar urea, this is 5 molar, 6 molar, 7, 8. Okay. Now what the urea page is doing is it is only breaking the interaction into a protein, right? So imagine that if this is the protein, right? It could be a single protein, or it could be multiple protein. Which means it could be a dimer, right? Or it could be a tetramer, right? So either of these conditions. So what you are going to see is, like for example, if we started the single at 0 urea, you are going to see a single band. Okay. Now, once it will enter into the 1 molar, right, there will be a change into this protein, right? So there will be a slightly opening. For example, if we adopt this kind of conformations. Now, once it adopt this kind of conformations, it is actually going to be of larger surface area. It is going to be adopt the larger surface area as it is keep going from the 1 molar urea to 8 molar urea. Okay, at 8 molar urea anyway, it is going to be completely extended. So that is the maximum surface area what this protein could actually be able to adopt. At the same time, when it goes from 1 molar to 8 molar, it actually can also be. So let's first discuss this. Okay, so if it is a monomer, right? For example, if it is a monomeric protein, it can actually adopt the different types of conformations, and these different conformations are going to be of different surface area or hydrodynamic surface area, right? Because as the protein is going to be keep unfolding, the surface area of the protein will keep going. So if the surface area of the protein will be keep going, the frictional forces are going to go up, right? Frictional is of this protein is going to go up. So imagine that if the 0 you are actually running like this, if the friction will go up, then it is actually going to run little slower, right? So you're going to have one band for corresponding to this one and the other band corresponding to this one in the beginning. But as it will keep running, so what will happen? This whole protein is going to be completely converted into this. So ultimately, for example, if this happened at 4 molar, then you are going to see that all the protein got converted into this, right? So that could possible, even if you suppose at 2 molar, it's possible that you will have the some of the native protein, some of the some of the protein which are of this one, and the sum of the protein of 2 molar, right? So even if you're getting the 3 bands, does not mean that that particular native protein is actually a trimer. It only says that this particular native protein is adopting the 3 different conformations which are corresponding to the 3 different amount of surface area. Okay, so because the when the protein is start unfolding, at some points it's also going to resist for the changes in its structure. So that's why it actually can form these kind of complexes, right? It could be trimer also. So if we talk about like tetramer, it will start with a tetramer, then it will going to one of the monomer is going to be broken, then it will come out, right? Like that also it's possible. It could be possible that all the 3 forms are coming from the monomer, and these 3 forms what you see is actually the 3 states of that particular protein. It could be oligomeric states, or it could be even within the single monomer also. These are the 3 states of the protein in terms of its surface area, not in terms of the oligomers. Is it clear, Abhi? Now we'll ask the others to if we have any more questions. So uh, Hemant, the final exam is going to be online at your center, like just like as it was been done the previous year also, right? Uh, Darpana, urea page is very much being used in the research labs on regular basis, and performing the urea page is very difficult. But what people do is not in here, but when I was in US, what people do is you can easily buy these gels actually, or you can buy the pre-casted urea page gel, and you can use that for conducting these kind of experiment. Otherwise, casting urea page is little difficult, but it is not uh, a very challenging thing. Once you do the multiple round, I think it is very much possible that you can be able to cast this. I think we have discussed how to do the casting, although we don't have the apparatus, so we cannot show you the demo. But I think it is possible that you can be able to cast this on a regular basis, and you can be able to conduct the experiments using the urea page. Okay, very good, Abhi. That I think I'm sure it's clear, right? That urea page is actually going to give you the detection of the folding intermediates. Those folding intermediates could be the different oligomeric status as well, but that is not, vice versa is not true. Okay. Rajesh is asking, sir, if we want to analyze a new protein without knowing its original charge, how the electrode condition in horizontal gel electrophoresis? If you are not know, if you don't know the charge, or if you don't know whether it is a basic protein or acidic protein, then we don't have any other option but to first do the in a horizontal gel. I think you are asking about the horizontal page, right? So in a horizontal page, it hardly matters, right? In a horizontal page, it doesn't matter because you can be able to. You don't need to know the charge. It will be run as per its charge. So if it is going to towards the anode or cathode, depending on whether it is positively or negatively charged. So as far as the horizontal page is concerned, I think it is not an issue. uh, Students, you should ask the queries if you have any more queries, because we only have very small time left, right? It's only for 1 hour. So if you have any questions, or if you could not be able to understand whatever we have discussed so far, then also you can ask me the questions. Uh, yeah, Abhi, I think the live sessions recording is also going to be available. So you can easily watch as many times you want later on also. And there's no issue. Avneet, you can ask me very simple basic immunology. I am not an immunologist anyway, so I don't claim that I'll give you the answers. But if possible, I think I will give you the answer. The Anshul is asking how to decide to stain Comasi blue or silver stain. It depends on the sensitivity, right? If you are working in the microgram range, you can easily use the Comasi because the Comasi is cheap and Comasi is safe, right, compared to the silver staining. But if you are working in the nanogram range or even lower than that, right, sub nanogram range, then the Comasi blue is not going to work. So then in that case, you can use the silver staining. Is it clear now, Anshul? Uh, so Darpana is asking, what is the Comasi blue, brilliant blue staining mechanism for staining the protein? Comasi brilliant blue is actually is a dye which actually goes and bind to the um, to the some of the amino acid side chains, and that interaction is non-covalent, right? It's not going to be covalently bind. Uh, so if you see the structure of the Comasi billion blue, what you will see that it is actually a positively charged dye. So that is actually going to bind to the some of the amino acids, especially the negatively charged amino acids present in the protein, and that's how it is actually going to stain the proteins through the interactions. Anshul, I'm not sure what kind of question you are asking, and Abhi also. Can you elaborate about this verification of AB complex structure by two approaches? What you want to discuss in that? It's important to know what your confusion is, so that because discussing the whole topic is not what we can do in this discussion sessions. Uh, we can just discuss the confusion or doubts what you have in. Abhi is asking why the 3D protein, 3D scaffold of the protein is not important for second approach. Um, the 3D structure is only important if the 3D structure is actually been involved into the interaction formation of the complex. But if suppose there is a small if there is a small portion or the only the the interaction is important, then and the 3D structure is not like playing any crucial role, then in that case I don't think the 3D scaffold is important, and I think that is what you have elaborated in your further write up, that urea is actually going to denature the three-dimensional structures, and that's how it is actually going to break the complex only if the three-dimensional structure is important. For example, if you have an enzyme and ligand, so enzyme and ligand are actually forming the complex not because they are related to each other by the complex, but they are also related to each other by the three-dimensional. Whereas in you can imagine the another scenario where a DNA is interacting with the protein. So DNA protein interaction is mostly been not governed by the three-dimensional conformation. It is always been governed by the positive and negative charges. So in that case, the 3D scaffold is not going to be important. It's going to be the surface charge which is going to play a crucial role. Then Hemant, I think you are asking that the SDS has two properties in electrophoresis, providing the negative charge and breaking the hydrogen bonds. So I think I'm not sure about whether it is breaking the hydrogen bonds because SDS is very hydrophobic. So I don't know if it can break the hydrogen bonding, but it is definitely providing the negative charge to all the proteins. Anshul, can you elaborate your questions? I think I'm not sure what you're asking. You're saying that how we can plot the graph for electrophoresis, which graph you're talking about, and what is your doubt? And Rajesh, how do we determine the required pH and volume for the reagents of our experiment? How do we determine the required pH and volume? No, I'm not. I'm not being able to follow your question. Uh, can you elaborate that? Anshul is asking in two-dimensional electrophoresis, which runs downwards. In two-dimensional electrophoresis, you are going to you can have many combinations, but the second dimension you are going to run the SDS page. So you in the first dimension, for example, although we have not discussed all the two-dimensional gel electrophoresis because of the time constraint and other kind of things. So what you see here is the two-dimensional gel, right? You can have the many properties. For example, you can run like the PI, right? That is standard, right? What you know, right? So if you run the PI in one direction, then in the second direction you can run the SDS page. So you're going to exploit in PI in one direction and charge in the other direction. Similarly, sometimes what people do is they actually isolate like in one dimension they run the things like ion exchange column, right? And the second dimension they run the SDS page or something like that, right? Uh, so then sometimes they what people do is they run the gel filtration, they run the urea page. Uh, so there's many combinations which are possible, and then followed by the SDS page. So that's many combinations of the two-dimensional electrophoresis which are possible actually. Okay, so we have only 5 minutes left more for concluding this session. If you have any questions, you can ask me. So, if we don't have any more questions, I am thankful to you for attending this live sessions, and I hope that the live session could have been very useful for you. And this is the first live session, so in case you could not be able to ask me the questions or you missed out, we can discuss again in the subsequent live sessions. So with this, we are going to conclude our live session now. Thanks for your interest and your attentions. Thank you.